introduce our today's presenter, I request Dr. Nikita.
Good afternoon and welcome to the translational series on today our guest speaker is Jeyashri R, a PhD scholar from Delhi under the guidance of Dr. Bhavana Prasher madam. Jeyashri ma'am will be guiding us on the topic gene expression and regulation. Ma'am,
Yeah.
you may please start.
Okay.
Yeah.
I'm audible, right?
So Dr.
Yes.
Jeyashri is a PhD scholar in CSIR-IGIB and her PhD guide is Dr. T.N. Vivek. She will tell us about her PhD topic also, and then she can move on with the presentation. Jeyashri, over to you.
Yeah. Thank you. So a very good afternoon to everyone who has been uh, attending this talk. And I'm Jayashri. I'm a PhD student currently in my final year working with Dr. Vivek T. Natarajan. So the theme of my lab is basically developmental biology, where we study the development of this particular cell type called melanocytes, which are actually involved in pigmentation. So the interesting aspect to these cell types is that these cells are actually derived from a population of cells called neural crest cells, which are basically present in the very early developmental stage as a multipotent population that can give rise to cells of all the three lineages like the ectoderm, endoderm, and mesoderm. So we try to understand how this particular cell type is developing from this multipotent population, and what are the cues, basically the genes that are necessary for driving the development of this particular cell type and not the other cell type. And that is exactly where the topic that I am covering today will also be playing a role, to be precise. So, with that introduction to my PhD topic, dwelling into the current topic for today, that is gene expression and regulation. So, we all have been like going through a few lectures to a few days for the past few days, where we all have understood how and what a genome is, and how what constitutes a genome, like basically what is a chromatin, what is a gene, and what are the sequences that constitute the expression of this particular gene. So, with that, we might be now familiar with the idea called the central dogma, which is basically the central theme that is involved in the flow of genetic information. So, we all know that the code or the information that is necessary for any cell to function is primarily encoded in the DNA, which is the deoxyribonucleic acid. So this DNA is present in the condensed form called chromatin, which then forms chromosomes and then present in a cell. And we all know eukaryotic cells have 2n number of chromosomes. Somatic cells basically have 2n cells, and the germ cells have only n number, that is only 23 chromosomes. So this DNA is what holds all the necessary information or the code that is necessary for the uh, genes that are necessary for the proteins to be formed. So now moving on from the DNA, it undergoes a process called transcription, which is where the polymerases called RNA polymerases binds to this DNA and then results in forming a structure called as RNA. So these RNAs are actually are the messenger molecules that carry the information from the DNA in the form of a molecule that can be read by the further machinery to form the functional molecules. These RNAs then undergo the process of translation with the help of ribosomal subunits to form the proteins, which are basically the polypeptides, which consider to be the final stage of this central dogma. So, as we know, but the interesting part is that we know how big the entire human genome or any genome for that matter is. But of this, only 2% of this particular entire genome constitutes this central dogma. That is where a DNA is transcribed to RNA to form a functional protein. What is the rest 98 percentage of the genome constituted by? That is basically constituted by these non-coding RNAs, which are considered to be the regulators. The coding RNAs are the messengers that results and follow this exact central dogma, but there are the rest 98 or the majority of the genome constitutes to these non-coding RNAs, which are considered to be the regulators of this coding segments, and this is considered to be, and this is the very like the current field of research where people are more interested in understanding how does this non-coding RNAs function and regulate the coding RNAs. So with this slide, we would be able to understand that the DNA holds the code, the mRNA acts as the messengers, and the proteins are the executors of the genetic information. So, with this very brief introduction as to what gene expression is and how does it flow in the cell, we can move on to the first process of this gene expression topic, which is transcription. So, as I have mentioned here, transcription is comprised of three stages basically, which is the initiation, elongation, and the termination process. So, as we can see in the schematic here towards the left, towards the right, we see that a particular DNA sequence has certain segments which are called as promoters and certain segments called as stop sequences and everything which is present in the nucleus. So the promoter sequence is basically specific to this group of enzymes called as RNA polymerases, which can bind to this particular region and then scan along the DNA sequence to form a to form a mRNA molecule in the 5' to 3' direction. So this so basically the genome is comprised of genes which are basically encoded in the coding strand and the non-coding strand. So the RNA polymerase scans through the non-coding strand to produce the RNA sequence which is complementary to the other strand to give rise to a mRNA which is actually similar to that of the coding strand in the genome. So now this particular mRNA is then transported from the nucleus to the cytoplasm to undergo some sort of post transcriptional modifications like a 5' capping, yeah, to undergo a 5' capping and a polyase tailing at the 3' end. So these post transcriptional modifications are very crucial for the mRNA to remain stable. So as we can mention here, as we can see here, it undergoes modifications like capping, then splicing to remove the intronic regions, and the poly-A tailing to form a functional mRNA molecule which will undergo further processing. So the DNA is transcribed not just to mRNA, but there are other groups of RNAs called as tRNAs, rRNAs, microRNAs, and piRNAs, which are basically non-coding RNAs. So how does this particular RNA mediate its function in the cell? So with continuation to the previous slide, here we can see the various classes of RNA molecules that are being transcribed from the genome. So mRNA is the most understood and the oldest group of molecules which have been there, and people have understood and studied it since the earlier times. So this is basically nothing but the messenger RNA which encodes for the proteins. Then comes the other classes of RNAs like tRNAs, which are transfer RNAs. So this tRNA and rRNA. rRNA is again the ribosomal RNA. These two group of molecules are very important for the next stage of gene expression, that is translation, because they are involved in synthesizing the functional protein machinery. So, and then followed by this snRNA and snORNA, which are small nuclear and small nucleolar RNAs. These small nuclear and nucleolar RNAs are very crucial RNAs for the process of splicing. So, they basically despite being RNAs, brings in other necessary factors or proteins to the mRNA molecule and help in cleaving the intronic regions, which are not the regions that is to be translated, and help in splicing or removing them from the mRNA to form a functional or a mature mRNA, which will undergo the further processing. Then comes the other group of small RNAs, which are microRNAs, and siRNAs, which are silencer RNAs. As the name suggests, microRNAs and silencer RNAs are very small RNAs. Basically, their length is only of 20 to 25 nucleotides. And these small RNAs are actually complementary or in sometimes are derived from the mRNAs itself, and then they can act complementary because they are derived from or complementary to the mRNA. They can bind to other regions, or they can bind either to the DNA or to the complementary mRNA, and helps in regulating the stability or the expression of the mRNAs. So basically, these miRNAs and siRNAs are considered to be decoys and scaffolds, which can actually help in deciding how whether the mRNA will proceed further for the processing or gets degraded. So basically, they are the regulators of the mRNAs. And long non-coding RNAs are LNC RNAs are basically long non-coding RNAs, which are similar to miRNAs in function, but their size is of greater than 200 nucleotides, which puts them under the class of LNC RNAs. And again, here they play the role of regulating gene expression by binding to mRNAs or to the genome itself, and help in bringing in factors to regulate or promote or repress the process of transcription. So with this is so RNA acts by being translated or by functioning directly as a RNA itself. So this is how the first step of gene expression, that is transcription, mediates or is being mediated in a cell. Then, to proceed further, the next step, which is the translation, that is basically the executor of the gene expression process. So now we have a mRNA, which is a messenger RNA, and these messenger RNAs have to undergo translation to form a protein. So here comes in the same processes as we have dealt with in transcription is being used here also, which is initiation, elongation of the peptides, and the termination. So, to put that into perspective, we see that the mRNA is bound by the subunits called as large ribosomal subunit and small ribosomal subunits, which is made composed mainly of this ribosomal RNAs, which I mentioned in my previous slide. So these subunits have certain sites called A, P, and E sites. The A stands for the amino acyl site, P stands for the peptide synthesis site, and E stands for the exit site. So as we can see, this particular, as the name suggests, tRNAs are bound to the A site and then brings in. So we know that tRNAs actually have, tRNAs are basically transfer RNAs which have amino acids at their 5 prime end, and in the loop region, which is here, they will have a stretch of 3 nucleotides, which is called as the anticodon. So you guys might have known what by now what codon stands for. A stretch of 3 nucleotides that encodes for an amino acid is called as a codon. So each codon has a complementary anticodon sequence which is present in the tRNA, and that is bound by a specific amino acids. So basically, we know there are 20 canonical amino acids which are specified by 61 codons, interpreted by about 31 distinct tRNAs. So basically, there are few amino acids which are encoded by multiple codons. So that is why we have about 31 tRNAs which encodes for a class of 61 codons basically. So this codon anticodon pairing will happen at the A site. And at this particular A site, when the first amino acid is being transferred, the peptidyl synthesis site, the P site, will help in creating the peptide bond between the two amino acids to form a dipeptide. So that dipeptide keeps on stretching as and when multiple tRNAs comes and keep on adding. So this process begins with the first tRNA, which is called as the initiator codon, that is basically methionine in all the cases. So generally, AUG, which is the codon for methionine, is considered to be the first amino acid present in the major class of proteins. So that is why this is basically a default step that happens with any protein, right? So with that, then the other nucleotides will keep on adding, and this process will continue until this particular A site encounters the stop codon. That is UAG, or there are a class of three codons which are considered to be the stop signal. So when this stop signal is being encountered, then the tRNA will exit. Then the incoming tRNAs will stop, and then the hydrolysis will happen such that the tRNAs dissociate from the peptide, and a functional and the peptide sequence will be present. So now this is not all about. So this is not a functional protein yet. So this is just a stretch of amino acids present in the cytoplasm. Now, how does it become so this translational pathway is actually mediated by a lot of molecules which are present. So the first step of this translation begins with, so now to delve deeper into this process of translation, here we can see we have the RNA which is bound by this factor. So there are a lot of factors with the respect to the steps itself. Like initiation is mediated by the initiation factors, the elongation mediated by the elongation factors, and termination mediated by the release factors. So here we can see that the first step of translation begins with this particular initiation molecule called as EIF2, which is bound to GTP and to the initiator codon, that is the methionine tRNA. So now when this forms the 40 along with the other initiation factors like 1A, 3 and 5, forms a complex which is called as the 43S pre-initiation complex. So now when this complex binds to the mRNA, it scans through the mRNA till it encounters the initiator codon, that is AUG, in the 5' to 3' direction. So when it encounters a start codon, then it triggers the recruitment of this large ribosomal subunit along with this other elongation factors for the peptide to elongate. So that only when the large ribosomal unit comes into picture, the A, P and E sites get activated, because of which the peptides are keeping on adding to the existing chain. Now, an interesting step that is mediated by this P site has a 28S rRNA, which is a ribosomal RNA, also acting as an enzyme which catalyzes the peptide bond formation. So the peptide bond is nothing but a bond that is present between the carboxyl group of one amino acid. So we know what an amino acid has, like it has a carboxyl group, amino terminus and everything. So the bond that is formed between the carboxyl group of one amino acid to the peptide or to the amino group of the amino acid in the A site is called, that is actually catalyzed by this particular ribozyme, which is the 28S rRNA. So, and when the ribosomal A site encounters a stop codon, no tRNA will be able to match it because it doesn't encode for an amino acid, because of which the release factor is being triggered to bind to this particular A site, which results in uncharging of the polypeptide, the ribosomal subunits, and the ERF complex, resulting in the termination process. So, this is the in-depth or the entire process that mediates the synthesis of a polypeptide sequence. So now when this polypeptide is synthesized, it is still a primary structure. The protein actually cannot function in its native form. It has to undergo a very crucial process called as folding. That folding process is what makes a primary structure of amino acid into a tertiary and a quaternary structure, which is a complicated or the complex structure of the protein necessary for it to function properly. So as we can see here, We all might know that how endoplasmic reticulums are classified as smooth and rough endoplasmic reticulums, right? So rough endoplasmic reticulums are where the ribosomes are present on them. So that is basically because when the ribosomes are present on the endoplasmic reticulum, they are basically involved in folding the proteins to form a functional protein molecule, which is can be then transported to different parts of the cell. So basically they are the factories where the proteins are actually synthesized and folded properly to be transported or delivered to different parts of the cell for executing the function, as which is mentioned here. But it is not possible for the cell to function without any errors, and it is possible that sometimes the proteins cannot fold properly due to various reasons, and that might result in misfolding. And obviously a misfolded protein is not functional, which can affect the protein's function. So those proteins, if they tend to be present in the cell, they can affect and become toxic. To avoid the toxicity mediated by such misfolded proteins, they have to be degraded, which is mediated by this degradation machinery, helping in maintaining the proteostasis. So this basically converts the polypeptide that is synthesized by translation into a functional molecule, that is a protein, which can be transported to other compartments of the cell like Golgi apparatus, and which will then take care in sorting them to different regions of the cell for carrying out their respective functions. So to put all this in perspective, the protein life cycle, as we can see in this particular slide, is called proteostasis, where the balance as homeostasis, which is considered to be the balance that is necessary for the cell to remain healthy. Proteostasis is a very specific term that is supposed to be uh, maintain the same balance in the protein formation and the degradation process. So basically, we know proteins are formed from amino acids. So this is a pool of amino acids which is being used for the process of translation to form a polypeptide, which is then folded to form a functional protein. Due to various stress factors like reactive oxygen species or stress induced by external agents like any drug usage or like whatever, there might be a lot of external factors. This results in aggregation or resulted in an unfolded protein, which might tend to aggregate. So, to prevent such aggregation and the protein from being dysfunctional, it undergoes degradation, which is mediated by this process of ubiquitin ligases. So basically, ubiquitin is kind of like, say, a signal or a mark that is to be present. So if a protein is not folded properly, then comes a signal called as this small molecule called ubiquitin gets tagged to those misfolded proteins by these ligase enzymes called E1, E2, and E3. This then triggers the proteasomal machinery to degrade the peptides marked with this particular signal, which indicates and present and helps in balancing the protein balance in the cell. Also, the folding is a very intricate process. So if you can see, this is a just a primary structure of the protein. So the amino acids have reactive groups present in them. So different amino acids have different groups like carboxyl groups, amino groups, and keto groups, sulfide groups, methyl groups. Everything is there. So now these groups are reactive among each other because they have a tendency to form bonds. That is the rule of nature, that any two reactive molecules present in close proximity can bind to each other through the formation of covalent bonds, or there are multiple types of bonds that can form. So that results in formation of such structures like alpha helix structures and sheets like beta sheets. And when these structures are then combined in multiple regions of the protein, it forms a tertiary structure. And when such tertiary structure comes together, it forms maybe a polypeptide, two polypeptides coming together. A protein is necessarily not always one protein functioning together. They can be like two proteins, polypeptides binding together, that might result in a functional molecule. So that is basically called as the quaternary protein structures. So this is how at the end a functional protein is either formed by folding, or the misfolded proteins degraded by the proteasomal machinery to maintain the intricate balance of the cell. So, proteostasis acts as a very critical checkpoint which ensures that only the properly folded and functional proteins execute their roles in the cell. So, moving ahead, now we have dealt with the process of gene expression in terms of how a gene from the genome is being transcribed to form an mRNA or other non-coding RNAs, and then undergoes either translation or they remain as a RNA and aids in the process of translation as non-coding RNAs to enable the gene expression and forms the functional entities, that is the proteins. So, but the question is the gene expression has to be regulated because we all know that a gene cannot be turned on all the time. The classic example for such scenarios is like we know women are basically they have two X chromosomes basically. So, if we can understand the chromosomes, they are called as alleles, right? So, those alleles have same genes present in both the chromosomes. But if the genes are going to get expressed from both the chromosomes, then it will be like a gene overexpression. That is not necessary for the cell. The gene has to get expressed only from one allele, right? For two X chromosomes at least, that has to be the case to balance the level of this X-linked gene product. So this condition is called as something dosage compensation. So this is one example of where a particular gene from a different allele has to be kept in check from being expressed. Next, very simple and a very and a scenario that we all can relate to is something like pigmentation. So we all know that people who are in tropical countries or who are exposed to higher UV tend to have a darker pigmentation. Like the pigmentation tends to be more, whereas people who are in areas of low UV exposure, they don't have that much of pigmentation. So basically, pigmentation is to protect our skin from the UV exposure. That acts as a shield basically. So that is naturally how it should be like. The tyrosinase is the gene which is involved in the synthesis of this particular pigment, that is melanin. And this particular gene has to be expressed in people with areas of high UV exposure. Whereas in people who are in areas where that much of UV exposure is not there, this gene need not get expressed. So it has to get regulated in such a way where it is influenced by the environmental factors, like the UV exposure decides whether a particular gene has to get expressed or not. So the genome has a very important decision of whether to express a particular gene or not. So how is this particular decision being taken by the cell? So that is when the transcriptional regulation comes into picture. So, what is transcriptional regulation? That is basically the basic key that decides a cell identity. So, we know every somatic cell in the body. So, we all begin as a single cell, right? Zygote, which is basically just a one cell, which then undergoes division to give rise to multiple other cell types, which then differentiate. But we all have the same genetic material, or this every cell has the same set of genes. But still, muscle cell is different from the neuronal cell, and a neuronal cell is different from say a melanocyte basically. What decides that this particular cell is a melanocyte, or this particular cell is a neuronal cell, or this particular cell is a muscle cell? That is basically encoded by what genes get expressed in each of the cell type. So in certain cells only certain genes will get expressed, that decides that this is going to be the identity of this particular cell. So how is this mediated? So the spatial and temporal. Spatial, it is basically the location, and the temporal, basically the timing. Both of these are very crucial factors to for a gene to get expressed. If a gene gets expressed at a time and place where it is not supposed to get expressed, then it is called gene dysregulation. So both of them has to be a very tightly regulated network to be functioning. So we know that the chromatin environment basically, so when a particular gene in a certain region of chromatin has to be expressed, it has to be loose enough for other factors like the polymerases to bind to them. So that is, they have to be in a euchromatin state. Whereas if that particular region need not be transcribed and be present as a, and to be repressed or kept repressed, then it present and it, then it will be in the form of a heterochromatin, right? So now, how does the heterochromatin and euchromatin gets decided? Like, what decides that this particular region of chromatin is chromosome is going to be in a heterochromatin form or a euchromatin form, which is basically mediated by this epigenetic modifications. So, as we can see here, chromosome, the DNA has a lot of regulatory regions called as promoter, enhancer, and there are other regions like silencer and inducers also, which plays the role of the as indicated by their names. So there are histones, which basically are the proteins around which the DNA is wrapped, right? So, and this histone morphology and how the histones are present will be deciding whether it is going to be a heterochromatin or a euchromatin. And what decides whether the histones will be open or closed is basically by these sort of modifications with respect to methylation and acetylation. So these post-transcriptional regulation of the histone proteins by acetylations and methylation molecules decides whether a particular region in the chromatin is going to be active or repressed for them to be transcribed. So basically, RNA polymerase again is not a specific entity. It can bind to any promoter region. Okay. So the specificity for the RNA polymerase is brought by a group of molecules called as transcription factors, which are the master regulators of the master regulators to decide whether a particular gene will get expressed or not. So, for example, say in the cell type called uh, melanocyte, there is a gene called as MITF, which is considered to be the master gene, which has to get expressed, and this particular MITF will get expressed only in this particular cell type and not in a neuronal cell. So that is, this MITF is a transcription factor specific to one particular cell type. So that is how the transcriptional regulation will help in deciding the or regulating the cell identity. So, this transcriptional regulation orchestrates the cell identity and the functions. So, with that, transcriptional regulation, this post-transcriptional regulation is as important as transcriptional regulation because mRNA is transcribed inside the nucleus, and then for it to even be transported from the nucleus to the cytoplasm, it needs certain cues, because without those cues, those RNAs cannot be bound by the transport machinery to be taken out from the nucleus to the cytoplasm for it to be translated, because translation machinery is present in the nucleus, in the cytoplasm, sorry. So for that to be functional, the post-transcriptional regulation is a very crucial process, which is mediated by non-coding RNAs, which are primarily microRNAs, LNC RNAs, and piRNAs. So, as we can see, the different post-transcriptional regulation machineries are the capping, which I have already form a mature mRNA. Addition of poly Addition of poly A tail, which is for the poly A binding proteins to bind and bring in the translation machinery, then deadenylases, and then other proteins which are necessary for them to get expressed or translated. So here,
Jayashri, just a minute. I think your slides have to be reloaded. Ayya Goa, is it visible? Slides are visible?
No, no, madam, no.
Yeah, so you will have to reload the slides.
Yeah,
Some
please,
interruption had
again,
happened.
you share your slide. In between, there was some connection error, and suddenly the
okay.
sharing is stopped. Please share it
Oh,
again.
is it visible now?
Yes,
Ah,
go to the
yes.
slideshow. Yeah, great.
Yeah, yeah. Yes, now it
Yeah.
is clear. Yeah, please.
Yes, sorry for the interruption. So, to proceed with the post transcriptional regulation part, the mRNAs, like the genomic counterparts, they also undergo some epitranscriptomic modifications like methylation of the RNA that actually is very crucial for to decide whether a mRNA will be stable and then undergo further processing by translation machinery. So these modifications are mediated by various enzymes uh, which are involved in transferring of methyl groups to these mRNAs and regulating their stability and further processing. So that this entire segment of post-transcriptional regulation helps in keeping a checkpoint as to whether, even though a mRNA can be formed, it necessarily need not undergo translation all the times, which when influenced by external factors, the mRNA can either undergo degradation or take the decision of undergoing translation. So that is basically put in check by this post-transcriptional regulation machinery, which is involved in fine-tuning the gene expression process. Now, with that, to proceed further, comes the next step, which is a very important process of translation regulation. So, we all know RNA to be a single stranded molecule. RNA is single stranded, unlike DNA, which is double stranded. So, but still, RNA being single stranded has secondary structures basically. So, as we can see here in this particular slide, there are regions in the RNA which are complementary to each other. Basically, the GC complementarity results in the RNA to bind to each other. The single strand can form such sort of a stem regions where the GC rich regions will bind to each other to form a stem region. And when there is some nucleotides which might not match with each other, and that might result in the bulge region to be present, and the regions where there is no complementarity, there will be a loop segment. So this sort of structures present in a single stranded RNA is called as RNA secondary structures. So this is a very important regulatory mechanism that is actually very it has been utilized in a lot of translational purposes as well, which I will deal with in a few minutes. So now, to understand how the secondary structure aids in translational regulation, here we can see a simple example of RNA which doesn't have any secondary structures, where there is a cap. As I have mentioned, a 5' cap is a post-transcriptional modification that happens that results in just a second. Yeah, that results in the 5' methyl guanine to be present in the 5' end, which is bound by the initiation factor 4E4G, which I have mentioned in the translational pathway process. So this will bind to the poly A binding proteins and recruits the small ribosomal subunit to form the pre-initiation complex, the 43S pre-initiation complex, which I have mentioned. So now this will scan through until it encounters the AUG and then recruits the large ribosomal subunits to undergo translation. So this is what is the very primary understanding that was there for mRNA translation. But this is a very primitive one. RNA do have secondary structures, and those secondary structures are actually very crucial in deciding whether the ribosomal subunit can bind or not. So, as we can see here, this presence of the stem and the loop region here in this particular RNA actually inhibits the ribosomal subunit from binding to the RNA, and then the translation can get inhibited because of this particular reason. So this is how the presence of secondary structure, sometimes which is mediated by external factors, can trigger the process or make the decision as to whether the particular mRNA has to or will undergo the process of translation, right? So with that, what happens is cap independent. So now this is all where the 5' cap is bound by some factors which helps in regulating or bringing in the ribosomal subunits. Now when there are some cases where the 5' cap is actually not involved, but rather such large ribosomal entry site structures, which is called as iris, which are internal ribosome entry site secondary structures present in RNA, will bind to the proteins poly A binding proteins and then recruits the ribosomal subunits to promote the process of translation. So this is basically used in a application perspective. If we viral RNAs are actually mostly translated by this particular process of forming large secondary structures. We know virus usually have RNA as their genetic material, and they use such machinery to exploit the host protein machinery to promote the translation of their own proteins inside the cell. So, blocking this particular machinery will help in combating the effects mediated by the viruses, is what is the application aspect of this RNA secondary structure. Okay. So now with this, here this particular figure summarizes how the RNA secondary structures have various applications and can regulate the transcription process, the post-transcriptional process, and can be exploited as drugs and vaccine purposes for combating the disease perspective as well. So this secondary structure of RNA is a very important property that helps in decoding the RNA to tune the protein synthesis process. So, a very classic example of this particular RNA secondary structure is this ribo switches. That is basically a very interesting system that is present in bacteria. So what happens here, as we can see, RNA, the secondary structure is what is called as a ribo switch. And this particular structure has the ability, like proteins, to bind to ligands, like metabolites, called lysine. So when lysine has to be synthesized in the particular cell, what happens is this lysine ribose switch is in the on state, and this particular RNA will not get cleaved because this cleavage sequence is not accessible to this enzyme. But when the lysine synthesis has happened for enough and there are excess of lysine, this metabolite can now bind to this particular region that results in this particular switch to get turned off here from this structure to this structure. So basically the structure changes depending on the binding of this metabolite. So that is why it is called as the on and off state, that is the ribose switches, which gets turned on and off depending on the availability of the metabolites. So this region which binds to the metabolites is basically the aptamers, are called aptamers, and in therapeutic purposes, these aptamers are basically short single-stranded nucleic acids which can bind small molecules and proteins, and they are being exploited in diagnostics, therapeutics, and in synthetic biology because they have very interesting roles because of their ability to bind such molecules. So this is one example of how the ribose switches that is present in nature in natural systems has been exploited for a translational perspective for drug development and is a very crucial for the gene expression regulation as well. So now with this in mind, the final and the last stage of gene expression regulation is the post-translational modifications. So now we have a functional protein which is folded and it is present in its specific location. But still, it need not function as it is supposed to function without these modifications. So that is when the role of these sort of chemical groups, peptides, or complex molecules like sugars and cleavage is necessary for them to function. So there are multiple checkpoints to ensure that from the genome to a protein, they have to be functional. They have to cross each and every checkpoint to form a functional entity, that is a protein which has these modifications. So the classic and the most understood ones are this phosphorylation and glycosylation, and this is mostly regulated by the intracellular and the extracellular environmental factors, right? So the phosphorylation, glycosylation are actually driven a lot by these factors, external factors to a greater extent, and they directly reflect on the signal transduction and the cell signaling pathways, and how the cells behave and interact with each other. So glycosylation is nothing but it's a process where glucose molecules, that is sugar moieties, are added to the proteins that is mediated by glycosyl transferases and glycosidases that is necessary for basically the proteins to remain stable, and that also gives them sort of a specificity as to where this protein will get localized. So different glycosylation decides different localization for the proteins. Okay. Now the next key player is phosphorylation. So we all know about those certain pathways that we all might have read is the MAPK signaling pathway, the ERK signaling pathways, which are actually well studied in cancer perspectives. Okay. So now these pathways, the key player is this particular gene called as phosphorylation, because the presence and absence of this phosphate group decides whether a particular protein is going to be functional or not. Some proteins, by addition of this phosphate group, will get activated, and some by removal or addition will get deactivated, and the vice versa is true. So this particular phosphorylation and glycosylation are basically interconnected PTMs that play a role, a very important role in regulating the cell signaling networks. So that ultimately decides how a cell is going to remain in homeostasis or not. So this perturbation might result in disease conditions, that is the most studied disease condition, that is cancer, basically. So now the next set of PTMs are the acetylation and methylation that are very important regulators of epigenetic regulators that helps in shaping the molecular memory. So why do I call it molecular memory is because here we can see zygote basically inherits two copies of each gene, one from the maternal and one from the paternal, that is the egg and sperm respectively. But both the copies of particular gene will not be active in the cell. There will be only one copy, either a maternal copy or the paternal copy of a gene will get expressed in a cell at any point of time. So how does the other copy remains inactive or suppressed, or why does only one copy gets activated? That is majorly because of these epigenetic modifiers, that is the DNA methyl transferases. So here we can see translational activity, that is where it is going to be, the gene is going to get expressed, and translational inactivity. So acetylation is generally considered to be a activation mark, where if acetyl group is present on histone, then it's the gene which is under the in that region will get expressed, and if a methyl group is going to be present, then that might result in the suppression of the expression of this particular gene. So this silencing or activation is achieved through DNA methylation and acetylation respectively in the histones, so that only the gene in one allele gets expressed and not in both the alleles. So that is how the molecular memory of what genes, so this that will follow through generations. Your mother will have certain thing, then the same will actually happen in the um, kid as well. Like what is the certain genes are maternally expressed and certain genes are paternally expressed, and that memory is encoded by this epigenetic modifications mediated by methylation and acetylation. So this is the next layer of regulation that is mediated by the post-translational modifications. So now this concludes as to how the gene expression begins and what sort of regulation or checkpoints are there to ensure that they get expressed at the right time and the right cell type without in proper structure and function. So this is the regulation that helps in deciding whether a gene gets expressed, and if it gets expressed, how does it function. So now, what happens if this particular balance gets dysregulated? What is the implications of when this is a very intricate process, right? So when that intricate process gets dysregulated, what happens? And the important question is as to why does it get dysregulated, and can it be treatable? So these are some questions that we think or people are trying to. So this is the basic biology. Till now we have dealt with the basic biology. Now the translational part of as to how can what happens if it gets dysregulated and how can we treat them is something that people are currently working on and which has a lot of scope. So with that thing, we see the most commonly studied dysregulation and the disease association is epigenetic dysregulation in cancer. So cancer is basically where cancer is basically nothing but hyperproliferation, where a tumor happens, like the cells proliferate uncontrollably, right? Why does it proliferate uncontrollably? Like why doesn't happen it in a normal cell? Because every cell can divide only to a certain extent, right? There are certain cells are terminally differentiated, that they cannot divide further, but that is not the case in cancer. They keep on dividing without any checkpoints. Why does that happen? Because there are certain genes, which are the proliferation genes, which have to be suppressed in a cell to keep the proliferation or division from happening. But because of this dysregulated epigenetic modifications, like activation of few genes where a methylation mark is supposed to be present, and activation mark being present results in activation of a gene called as c-MYC. c-MYC is basically a proliferative oncogene basically. So, and cyclin D1 gene. So when these genes are overexpressed, or if their expression is not kept under check, then they keep on getting expressed, results in overexpression, ultimately resulting in the cells proliferating to a greater extent. So this is one case, and this is the major chromatin remodeling. Then follows by how this particular methylases and enzymes not being functional, resulting in the lack of epigenetic marks, resulting in overexpression of gene, or in some cases expression of non-coding RNAs. Certain non-coding RNAs have to get expressed to keep the expression of certain coding RNAs in check, or to prevent them from getting expressed. But when their expression gets dysregulated, that in turn affects the expression of the coding regions or the coding genes, that results in the disease condition. So, this is how the epigenetic modifications in the genome or in the proteins associated with the genome results in the disease conditions like cancer. So this summarizes what I have told in this particular thing, where abnormal methylation or reduced acetylation results in a lot of tumor conditions in general. With that, and another disease condition or an example is that, where here we can see this is basically the eye, a disease condition called as glaucoma, where basically people lose their vision because of pressure buildup in the eye, where the neurons get the cells, the retinal ganglion cells here basically starts to degenerate because of pressure, and that results in the vision loss process. So, at a molecular level, what exactly is happening is that the expression of genes like P300, P53, which is a tumor suppressor gene, all has to be activated in this cell for them to be functional. But because of HDAC gene overexpression, which is a histone deacetylase, so this enzyme, what it does is remove the acetyl group. So removal of acetylation means suppression of gene expression. So these genes might not get expressed when actually they are supposed to get expressed, because of which they might result in this particular cell getting damaged. But people have used pharmacological molecules like HDAC inhibitors, which will help in inhibiting this particular enzyme, that will help in restoring the expression of these genes, and then promotes the survival of this particular neurons, and can help in combating this particular disease. So this is how these are the implications of what happens when the gene expression regulation machinery is getting dysregulated. So now, what are the causes? Like, what induces this particular gene dysregulation is an important question. So there are a lot of factors. Like, as I mentioned in my example to tell about how UV exposure decides whether a particular gene is getting expressed or not, which is an environmental factor, right? So here we have factors which can be contributed by the climate, which can be contributed by the presence of pollution, presence of toxins in your environment, that might actually play a very critical role on these epigenetic modifications. Because These, as I mentioned, if a pharmacological molecule can inhibit your histone deacetylase and function in a certain way, unknowingly, presence of few toxins in your environment might cause some changes in human beings, and that might result in your epigenetic machinery getting dysregulated, which might result in this sort of developmental disorders. Also, you might know about various genetic alterations like point mutations, inversions, deletions, transversions, and translocations happening in the chromosome that might also mediate these sort of gene dysregulation, resulting in a lot of disorders, primarily neurodevelopmental disorders, carcinogenesis, which is tumors in various organs and various tissue cell types, and gene tech. It's basically a very multifactorial complex process, and the predominant causes are basically these genetic alterations, environmental factors, and stress. Also, in recently, people have started to understand as to how the stress levels, like how the link between the gut brain barrier, the gut brain link is something that people are currently trying to understand to a great extent, right? So how the kind of food intake or the how the gut health actually decides various disease condition is something that people are trying to understand, because the microbiota present in your gut actually play a major role, other than just taking care of your digestion process. So these sort of factors, that a lot of external factors actually help in deciding how a gene regulation actually is kept in check and what happens if it gets dysregulated. So, with that, how to combat this thing? Now, this is a very a recent and a new group. It's a very upcoming field of research where people are trying to understand if these gene dysregulation can be combated or by taken care of by various strategies like gene therapies, or by using inhibitor molecules against various enzymes and genes. And how can the structure or the RNA secondary structure, which I already mentioned while dealing with the translational regulation part, can be exploited for the development of molecules that can be useful for treating disease conditions. So, as I have mentioned before, so the targeting these molecular checkpoints will pave the way for development of precise and effective therapies to combat gene dysregulation that helps in regulating the gene expression and bringing the homeostasis back to normal, right? So, with this, I would like to conclude my talk. So, these are some of the references which you can look for. There are some textbooks which you can refer for understanding these concepts in more in depth, and some articles which you can refer to for to understand the current developments and research that is being carried out in this particular field, right? Uh, so with that, I have some few questions. So should we take this now or later?
You can post it in the classroom, and
Okay.
then yeah, at the end maybe the organizers would like to send them to the participants, especially the student participants, so that they can recap the whole class and all that.
Yeah, sure.
Yeah. So thank you, Jayashri. Just one clarification, as you mentioned that one copy of gene is always expressed. Is it also true for the adult life? I think the genomic imprinting to keep the other copy untranslated is for a specific set of genes. It's not for all the genes. Am I right?
Yeah. Yes, yes, yes.
So the genomic imprinting is for specific set of genes that keeps the other copy silent. That's what
Yes.
yeah. So that's what something I thought I should make it, and it's a very elaborate presentation. If there are any questions, please put it in the chat box.
Yeah.
Dr. Jayashri, one question is there in the chat box. Are you able to
Yeah.
see, or shall I read?
I'm not able to see the question.
Uh,
Oh, yeah, I got it. Yeah. Prakriti has influence on DNA-directed protein synthesis. Wait. Okay. So I am not a Ayurveda person, so I really don't know exactly like how can this influence. But based on my discussion with my peers and my colleagues, I do think that an IGIB has been carrying out some pioneering studies in this field of genomics, where they are trying to relate how prakriti types can influence the expression of certain genes and regulate the development of certain genes. So yes, it do have an influence on DNA directed, but this very specific process of DNA directed protein synthesis rate is something that I'm not sure of. But yeah, it does have an influence on expression of certain genes, is what I understand. Yeah. As Ayurveda students, we sometimes find it difficult to see clearly how these concepts connect with our science. Apply this knowledge within the Ayurveda framework. Okay.
Yeah, so I'll take that question, Jayashri. So it's actually not that everything according to, so all processes will change in different prakriti. That's not the case. The biology will function as the biology has to function. From this DNA to RNA to protein and protein regulation, it has to happen the way it happens in biology. But why we study all these things? To know if any of these processes or regulation is different in any different disease condition, and whether that difference has to be corrected in some way or the other. As Jayashri also mentioned some examples about small molecule inhibitors of HDAC, or things which will change the RNA structure to be able to regulate the translation process also. So if we know how in biology things are functioning and how in molecular biology things are getting regulated, all these regulations happen either in response to the internal environmental changes or in response to the external environment. And same Ayurveda talks about environmental influence on Vat, Pitta, and Kapha, or internal biological differences in the biological processes of Vat, Pitta, and Kapha. So the question is whether we understand this as it is, and we understand our Vat, Pitta, Kapha biology as it is, and then we try to see whether, if we want to see the difference, we will have to find Vat, Pitta, Kapha models, as I said in the first class also, and see if these subtle differences are visible in different Prakriti individuals, like in case of DNA and protein. So DNA directed protein synthesis rate is a very specific problem, a specific question. But if I were to answer that, if different healthy individuals of the same age group, of the same geoclimatic condition, more or less same dietary practices, still, and also healthy, they are not suffering from any disease, but if they were, when they were examined for any molecular changes in the level of gene expression, we do find baseline patterns of gene expression differences amongst Vat, Pitta, and Kapha. Now, if we were to repeatedly observe this, or if we were not to do even repeated observations, but whatever observations we made, if we can follow it up at other or levels of organization, like protein level or biochemical level, or even at the so if we were to validate it at different biological levels, or we observe the same patterns in differences in Vat, Pitta, and Kapha, then we can say that when it is, although the biology functions the way it is, but certain genes are more expressed in certain prakriti, because the relative proteins will be more expressed, and the functions will also be seen to be highly expressed. One such example is that of a cell cycle related genes more expressed, or cell cycle and DNA repair genes more expressed in Vata individuals than the other two. And at the cell line level also, we see that the proliferation rate also differs between Vata cell line versus Vata LCL versus Pitta LCL and Kapha LCL. So this is how, so first we understand where, how it functions, how it gets regulated, what are the factors which influence the regulation, what are the factors which influence the differential expression of proteins as well as biochemical pathways. And then we try to find whether those differences can be seen in Prakriti, and whether that can be then assigned to a dosha. So we will have to go systematically in terms of understanding, correlating at a conceptual level, and then designing the specific experiments with an hypothesis to test whether what we are thinking is true or not. That's how we proceed. And in the last lecture on 17th of October, we will discuss some of the examples of the studies we have done or other groups have done to see how we are able to make the bridge between dosha, prakriti, and molecular biological differences. Or first of all, what is the basic way in which the cell functions? Then look at the differences in which differences which it produces in different conditions, and whether those differences are viable differences to be called as within the healthy limits, and when it goes beyond a limit, whether they can be considered as a disease conditions. So that's how we will go systematically in understanding and keep discussing, have more brainstormings, think about how we can correlate and what are the things that have already been done, what next can be done. So that's the way we move in science.
Think one last question where they have asked about how entropy within gene regulatory network considered as early indicator of disturbances even before changes in gene expression levels become visible. So to take this entropy question, so I also have been recently working on something similar where we have some data where we were looking at how differentiated a cell type is. So entropy is basically, I think, to my understanding, is basically is the randomness or how variable is an environment in a cell. So that basically, I think the question that you have asked is like other way around. Entropy is basically decided by how diverse or how variable your cell type is. So basically, a low entropy means your cell has very restricted or a very specific set of genes getting expressed, whereas a cell with high entropy will have a very large group of genes getting expressed. So I think the gene expression decides, or the expression of genes decides the entropy of the cell, is what my understanding is. So there are some parameters like Shannon entropy and all, which methods even I am exploring, so I am not very aware of the computational approaches, but Shannon entropy is one such thing that people look into when they want to understand how a pluripotent or stem like a cell is compared to how differentiated it is. So yeah, that is something that I can answer for this particular question. So I think no more questions. Take low entropy as a homeostasis like prakriti state. Yes, I think we can maybe, if you want to consider homeostasis as low entropy, yeah, if you can comparing two states like a disease versus a normal state, then maybe. But finding entropy difference with between different prakriti states is something that I'm not very sure how much of a difference we will be able to find, because if prakriti is known to uh, decide on those things, only then the entropy changes will come about with prakriti. So that correlation is something that needs to be tested out. Yeah, right now if there are any studies, that I'm not aware of. Methods for assessing dysregulations. So that they are standard molecular biology techniques like high throughput. There are a lot of high throughput techniques like sequencing, RNA sequencing, whole genome sequencing to assess the genomic alterations that people do, exon sequencing, and so yeah, sequencing strategies have now been like there are spatial transcriptomics are being done at this moment. So yeah, sequencing is the way to go about it.
And moreover, for assessing the dysregulation, you will have to have a serial sample perhaps, or comparison of two groups to be able to understand. So, or you will have to have previous state and how dysregulated it is getting in response to any external trigger.
As there are no more questions in the chat box, ma'am, I would like to thank you, Dr. Bhavana Prasher, madam, and Jeyashri, ma'am. As we belong to the faculty of Ayurveda, this topic are very new to us. I would like to thank you for making it easy for our understanding and with your effort and time. Thank you so much, ma'am.
Okay.
Thank you.
Thank you.
Thank you, madam. Thank you so much.